Hello everyone, welcome to today's presentation entitled Population Scale Human Genome Analysis on the Cloud. My name is Peter White. I'm Director of the Biomedical Genomics Core here at Nationwide Children's Hospital in Columbus, Ohio. I'm also an Assistant Professor of Pediatrics at The Ohio State University. And I'll be speaking today with James Hermes, who's CEO of Genome Next. Our presentation today will be in two parts. We'll be discussing the work that we did collaboratively with my team here at Nationwide Children's and the team at Genome Next. But before we begin, I just have a couple of announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the QA box, which can be found by clicking on the green QA button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. And if you have any technical problems viewing or hearing this presentation, please click the support button at the top right of your presentation window, or submit your problem through the green QA button on the lower left. Finally, I do want to thank LabRoots for their work in putting this conference together. And especially we want to thank Intel for sponsoring this presentation. So with that, I'll go ahead and get started. So the Human Genome Project began in 1991, was an international effort to sequence the first human genome. And it took an estimated 15 years to complete. With a cost of about $3 billion. So that's a dollar for each base pair in the human genome. Now, if you want to get an idea of what it would look like if we were to print the human genome, these photographs are taken from the first printed copy of a human genome that's housed in the Wellcome collection in London. And as you can see on this bookcase, it contains over 100 volumes with just each volume contains all of these pages with A, C's, G's, and T's, the 3 billion characters that go to make up the genome. So fortunately today, we're able to sequence a genome a lot faster, and you don't need an international consortium of scientists to sequence. These are two of the sequencers that we have in my lab here in the Research Institute at Nationwide Children's. The smaller machine on your right is the Illumina MiSeq. This is an instrument that we use for bacterial genome sequencing and for smaller sequencing studies. But our main workhorse over the last 4 years has been the Illumina HiSeq instrument. This is capable of generating 3 billion sequencing reactions in parallel, roughly producing about 1.2 terabytes of raw data in a run. And this allows us to sequence a human genome in as little as 40 hours. So a focus of my research has been on dealing with all of the enormous amounts of data that this instrument has been generating. We've sequenced thousands of samples. We have servers with trillions of base pairs of data. And so the research of my group has been in an area called translational bioinformatics, where we try to take this data and turn it into something meaningful in terms of molecular diagnostics, clinical treatments, and clinical outcomes. Now, it would be nice if the output from these sequencers actually looked like what we see on the bookcase here, but it doesn't. The sequencers are only able to sequence the genome in small fragments. We go through a process in the lab where we shear the genome down to about 300 base pair fragments, and then we're able to generate anywhere from around 100 to 150 base pair reads from each end of that fragment. So it's a bit much like if we were to take this entire collection and run it through a paper shredder, getting output like this. When we first started using this sequencer, I think this was our reaction to the volumes of data that were coming off of it. Because what we needed to do is go through a complex analysis process of taking all of this output and reassembling it to get back to this original image. And in the case of pediatrics, where we're often sequencing kids with rare genetic disorders, what we're looking for is the one page in this entire collection that contains a typo. Where is that one variant within a gene that could be causing a disease? And here you can see it in red. We have a C that's actually on this page, a T. And so this is what the process that we're doing in terms of analysis is going from that raw output to finding this potentially pathogenic variant. So to do that, there's a number of steps in the analysis workflow. It starts on the instrument itself. The sequencing process generates these images that look like this star cluster at the top of the screen there. And the first phase of analysis we call primary analysis. This is really taking those images and turning them into intensities and base calls. Subsequent to primary analysis, once we have those base calls, we can begin the process of secondary analysis. And this is where we go through a number of steps to actually determine what given variants a patient may have. And then finally, tertiary analysis is the more advanced bioinformatics, actually interpreting what those genetic variants are. But for a long time, the bottleneck has been in this area of secondary analysis, going from that raw data to a list of genetic variants. So the process to do secondary analysis encompasses these 5 steps that I'm showing you here. It begins with the raw output from the instrument, which is in FASTQ format, and that has to go through a process of alignment back to the reference genome. Then there's a stage of that's called a local realignment that corrects for alignment errors that may be associated with small insertions and deletions in the sequence. Then we remove any duplicate reads that were introduced during the laboratory phase of library preparation prior to sequencing. And the next step is to recalibrate the quality scores that are associated with each base, and that allows us to more accurately perform the final phase, which is variant calling and genotyping. The final output of this process is what we call a variant call format file, or a VCF file. And this contains all of the variants for a given sample. And for a whole human genome, that's typically in the order of about 4 million genetic variants. So the problem that we ran into when we first started doing this was that we had an instrument that was able to sequence a human genome in, as I said, about just under 2 days, 40 hours, but it was taking us 2 weeks to do the data analysis. And our solution to that was this, to come up with a new way of performing the analysis by carefully examining each of these steps. We optimized the process by which the analysis could be distributed across multiple computers and CPUs within a given computer. If you were in John Quackenbush's session earlier today, he said that bioinformaticians like to name their software after cute animals, and he was describing an algorithm called Panda. We named our algorithm Churchill, and I guess looking at Churchill there, he's not such a cute animal, but he's certainly a name that has helped people remember. It sticks in people's mind when they hear the name of the algorithm. So I'm not going to go into great details about how we did the processing of each of these steps, but it is described in full detail in a publication that came out earlier this year in Genome Biology. That's an open access article that you can easily download. So when we look at this process of doing the secondary analysis, there's a couple of different approaches that can be taken to parallelization. One approach was to parallelize by chromosome. But as I'm showing you in this slide, there's a limitation to that approach. It's a natural way that the genome can be divided up. We have the 22 autosomes and the 2 sex chromosomes here, so in total we've got 24 chromosomes that the analysis process could be divided by. But there's 2 limitations. The first is that you're limited to 24. If you want to scale beyond 24, you can't, because that's the total number of chromosomes that are available. The second limitation is that of chromosomal size. You'll see that chromosomes differ in size. Chromosome 1, being the largest, is about 250 megabases, and chromosome 21, being the smallest, is around 50 megabases. So any analysis that's parallelized by chromosome will be limited by the size of the largest chromosome. Chromosome 21 will finish first, and chromosome 1 will be the last to complete analysis. So what we did with Churchill is we came up with a strategy to balance the parallelization by dividing the genome into subchromosomal regions. And what this is showing you here is this balance by dividing the genome up across here. In this example, it's 24 CPUs to do the analysis. You can see that each processor is processing the same amount of data, and thereby the analysis has become balanced and more efficient. And that's what we observed, that When we compared the parallelization strategy utilized by Churchill with two other pipelines, a pipeline called HugeSeq that was developed by Stanford that uses chromosomal parallelization, and then a pipeline from the Broad Institute called GATK-Q that uses scatter-gather parallelization. And what these plots show you are utilization of CPU for a single server, going from 0 to 100%. And what you can see is that Churchill had the highest utilization at about 92%, followed by HugeSeq at 46% and GATK-Q at 30%. So the other advantage of our approach was that, one thing I want to point out here is that you'll see that the chromosomal parallelization approach used by HugeSeq, the utilization plot closely matches that of the size distribution of the chromosomes. So what's happening is chromosome 1 is taking the longest, whereas as the smaller chromosomes finish first, your resource utilization declines, closely matching that pattern. So what we observed with the result of the balanced parallelization strategy utilized by Churchill was that it was highly scalable. So this plot shows you the amount of speed up you get in terms of the analysis time as you increase the number of CPUs. And what we observed is that both HugeSeq and GATK-Q really didn't scale well beyond 48 cores, whereas Churchill continued to speed up as we used more cores for the analysis. We did a computational prediction using Amdahl's law on what the theoretical maximum speed up you could get, and we observed that Churchill tracked closely with that predicted by Amdahl's law. The result is that we could drastically reduce that time frame down from that 2 weeks down to under 2 hours to do the analysis. What I'm showing you here from on our in-house server with up to 192 cores, we're able to reduce the analysis time down to just under 5 hours. And then when we ran on a larger cluster at the Ohio Supercomputing Center with 768 cores, we reduced the time down to just under 100 minutes. It's recent work in my group, we've been able to, using Amazon Web Services, we've been able to reduce this time frame down to 77 minutes. And one of our goals is to get this to under an hour within the next few months. But we work in a pediatric institute that's currently using Churchill through Genome Next clinically. And one of our goals was not only to increase the speed of analysis, because that can be a matter of life and death for kids with rare pediatric diseases, but we wanted to ensure that we didn't make any sacrifices to the quality of the data. And using the National Institute of Standards and Technology's Genomes in a Bottle sample that was a reference that was released last year, we were able to see that across the board, Churchill had the highest rates in terms of validation of known variants. It had the highest accuracy and lowest error rates. In terms of the overall diagnostic effectiveness, which is measured by the Uden index, which is the sum of sensitivity and specificity -1, we were seeing data at 99.66%, so again the highest diagnostic effectiveness. But the other advantage of our approach for parallelization is that it's 100% reproducible. Every time we run the analysis, we get the same results. And it's also deterministic. And what I mean by that is, regardless of the computer system the analysis is run on, or regardless of the level of parallelization, if you're using 24 chromosomal subregion or you're using 1,000 chromosomal subregions, you get the same results. The final VCF will be the same. And we believe this is the only pipeline that's able to make that claim of both reproducibility and determinism. So how does this relate to population scale genomics? Well, one of the things that we realized, while Churchill was very fast for an individual sample, we believed that it could scale effectively to do much larger scale studies using thousands of samples. And why this is relevant is this is a picture of our sequencer in the lab that currently can sequence, if we ran it 24/7 back to back, we could do at most 150 genomes per year. And the cost of that would be pretty prohibitive at about $5,000 per genome. But Illumina announced last year new technology that would enable population scale genomic sequencing. This is a photograph of the HiSeq X instrument that's able to sequence up to 18,000 genomes per year at a cost of roughly $1,000 per genome, and generating enormous volumes of data at 1.8 petabytes. And what we're seeing is that there are a number of studies occurring around the globe where nations are sequencing their populations. And this is a photograph of the cover of this month's Nature Genetics, where Decode Genetics in Iceland published the results of a study of 100,000 Icelanders. In my home country, in the United Kingdom, Genomics England is a study funded by the National Health Service that plans to sequence 100,000 patients. And then also the Saudi Human Genome Program, again targeting to sequence 100,000 Saudi genomes. And then the Faroe Genome Project is the Faroe Islands plan to sequence their entire nation, which is about 50,000 people. So we're starting to see population scale studies beginning. So what we did last year is we proposed a proof of concept study to Intel through something that was called the Intel Head in the Cloud Challenge on Amazon Web Services. And what they did is they solicited ideas from groups that wanted to use the servers on Amazon, on AWS, that are powered by Intel processors, to do an analysis that was of a large scale. So along with our partners GenomeNext, we proposed a study where we would do analysis of the 1,000 genomes data, which is one of the largest publicly available data sets. What the 1,000 Genomes Project is, it began in 2008 and has been the work of an international consortium of over 400 scientists to create the first deep catalogue of human genetic variation. And in October 2001, phase 1 was completed, and the group released the population variant frequencies for 1,092 individuals. And then in April 2013, the group announced that they had completed their sequencing phase. And then 18 months later, in September 2014, the final variant calls and population frequencies were released. And this was for a total of 2,504 individuals across the 26 populations that I'm showing in the map below. Now, the great thing about the work that the 1,000 Genomes Consortium has done is they've made all of this data publicly available. All of the raw sequencing data is stored on Amazon's it's stored in an Amazon S3 bucket, enabling groups to access it and do their own analysis. So the first thing we needed to determine was to show the Churchill would work on AWS. And so again, this is showing you a resource utilization plot similar to what I showed you earlier. And we're seeing that we had highly efficient utilization of this is a single EC2 instance, enabling us to complete analysis of a 30x human genome in about 11 hours. Then as we increase the number of instances, the graph on the right shows you how we're able to reduce that time frame down to under 2 hours by the time we get to 16 instances. And in this case, we actually used a more computationally intensive algorithm to do the analysis that was recently developed by the Broad Institute called GATK Haplotype Caller. Using the Churchill parallelization approach, we're able to perform whole genome haplotype calling in about 24 minutes. So given that we could show it would work for a single sample, we then went about designing how we would do this analysis for the 1,000 genomes data set. Now, as I said, it's all stored on Amazon in an Amazon S3 bucket. All 2,504 individuals were sequenced with two different methods, whole genome sequencing and whole exome sequencing. So we actually had in total 5,008 input data sets. We're able to parallelize the analysis of that using Churchill to parallelize that through the FASTQ to BAM step of doing alignment, deduplication, indel realignment, and recalibration. And the final output of that was for each genome, each sample, we divided them into 512 genomic subregions. This gave us a total of 2.6 million parallel processes during this phase. Now, the second step was to take these BAM files, these process BAM files, through the process of genotyping. And we did this using a format called GVCF, or genomic VCF genotyping. And so for each sample across the 512 regions, we were able to do variant calling in parallel, again about 1.3 million parallel processes during this phase. The output of this was each sample had a GVCF file that was divided into these 512 subregions per individual. The final step was to take these GVCF files and do joint variant calling on them. So for each region, we could take all 2,504 individuals and perform the variant calling that would ultimately produce the population allele frequencies across all of the samples. So we were able to do this from FASTQ, those final population frequencies, in 7 days. And what we found is that we had very high level of overlap with the variants that were called by the Thousand Genomes Consortium, as shown by the top Venn diagram. But we did observe that we discovered about 5.6 million novel indels that were not called by the Thousand Genomes Consortium. And we believe this may be that potentially we used a more sensitive approach to do the genotyping by using this GVCF format along with haplotype caller. But what we did see is that for the variants that we called in common, there was an extremely high level of correlation in terms of the frequencies that they reported and then the frequencies that we observed. So to summarize this part of the talk, what we were able to do is, using Churchill, we were able to perform genomic analysis of an entire population of 2,504 samples. This was about 148,000 input files, around 68 terabytes of compressed data, totaling 85 trillion nucleotides. We did this analysis using a haplotype-aware genotyper, and were able to perform joint sample calling. And the final results closely correlated with that of the Thousand Genomes group. And we're able to do all of this in 7 days. So I just want to end this part of the talk by thanking the members of my team here at Nationwide Children's, especially Ben Kelly, who's one of the lead developers on Churchill, and James Fitch, who is the lead developer on the Thousand Genome study. And this work has been supported through our partners at GenomeNext, along with Amazon Web Services and Intel for funding the Thousand Genome study. So with that, I'm going to hand over to James, and James Hermes is CEO of GenomeNext, and he's going to describe how we actually did this from the cloud computing perspective.
Thank you, Peter. This is James Hermes with GenomeNext, and I'm happy to be here to present the GenomeNext platform and how we supported Nationwide Children's Hospital on the Intel project ahead in the Cloud Challenge. So I would like to say that as we go to the presentation, please feel free to, there's a Q&A section at the end. We're going to try to devote about 10 minutes of time. So if you do have any questions, please feel free to post those in the Q&A. So in terms of the GenomeNext mission, GenomeNext was formed as a bioinformatics company to accelerate the promise and the capabilities of precision medicine and scientific discovery. As part of that mission, we looked at the business problem that is currently prohibiting users from doing genomic analysis. And a lot of that has to do with the automation and the ability to simplify the overall workflows for the secondary and tertiary analysis. So part of our mission is to automate that from beginning to end, from the sequencer all the way through variant calling to the annotation. Additionally, one of the challenges that we have seen with organizations is the ability to scale on the technology on the side for the infrastructure. And so one of the things that GenomeNext is solving is the ability to use cloud computing environments to remove the limitations at a local level in terms of storage and compute. And for that, we're actually working in partnership with Intel and Amazon Web Services, and we use the Amazon Compute Cloud to build a software as a service that performs genomic analysis. So to look at the overall platform, so we built the software as a service on top of the Amazon environment. And that software as a service is layered into multiple aspects. One is we have a portal user interface. We handle the authentication, the content management, the automation of the cloud infrastructure, as well as all of the analysis pipelines that are used in terms for the genomic analysis process. Additionally, the bottom layer is we deploy a security operations center to maintain the logging, web application firewalls, and the overall compliance protection of the environment. So from a security and compliance perspective, we spent a lot of time ensuring that HIPAA compliant on Amazon's cloud, that we also were able to work with CAP and CLIA processes to incorporate those within organizations. Additionally, data upload to the cloud is something that we've incorporated into the environment to simplify the large data upload, and we'll talk a little bit more about that as we move forward. In terms of content management, we built a light content management that allows for teams, projects, samples, and analysis requests to be managed at an enterprise level. We additionally employ security trimming so that only certain users have access depending on your organizational needs. The cloud infrastructure management, which is probably the thing that's going to pertain the most to what's novel about GenomeNext, is the ability to launch and manage on-demand thousands of Amazon instances or servers, as well as the amount of storage and the automation that brings all those things together. So we are able to manage that and provide that as an elastic on-demand service. And then we have taken the Churchill pipeline, and we have been able to take that pipeline and fully automate it onto the platform. So a user can upload data, request an analysis, submit the analysis, and for a whole genome, within a couple hours, that analysis results come back to that user. So the Intel Head in the Clouds Challenge, Peter summarized it very well, but essentially we were looking at 2,504 individuals across 26 populations. Obviously, we're talking about a lot of data, 70 terabytes of FASTQ data, and lots of FASTQ files. And in the end, we were looking at managing and running close to 1,000 EC2 instances over a 7-day period. And being able to manage that complex workflow from taking FASTQ files, creating samples, and running analysis requests and managing the infrastructure is really the problem, the large data problem that we were solving on Amazon. So the first challenge is really a challenge of data upload. So how are you going to get 70 terabytes of data from one location to our cloud environment? And how are you going to do that efficiently and that it's not going to take a lot of time? So one of the advantages that we had is that our cloud infrastructure, our software as a service, runs on Amazon, is co-located in the same region as the storage of the phase 3 data. And that is in the US East Coast, or the East region. And so deploying GenomeNext in the same region near the data set allowed us to be able to reduce our latency and the time that it would take to move that large data set across. And that phase 3 data is hosted by Amazon, and it's open to the public, and therefore it allowed us to automate the data upload from their S3 storage to pull those FASTQ files and automate the creation of the samples, and then eventually to migrate the sample data to our EC2 instances to do the analysis and drop the data back. To do that, we had to make sure that we were running enhanced networking performance, 10 gigabit per second on the different EC2 instances that we were running. So this gives you a little bit of a view from a user interface of the data upload solution. So there's options where you can create samples, you can upload data. We have an uploader that can upload one sample at a time. However, that wasn't going to be sufficient for this project. So what we developed for this project was the ability to upload an index of files that map to the S3 bucket location where the phase 3 data existed. In doing so, we could upload the index, and that index had all the mappings to the data. And when we did that, we were able to automate the creation of the samples that were used for the project. So the second challenge is really around scale. So the question here is, we essentially are working with 5,008 genomic samples. How are you going to efficiently scale up your infrastructure and move the data across and launch thousands of instances in order to run analysis? You have to do that efficiently. You need to take security into account. You also need to take into account all the different processes and monitoring that has to happen. So from a very high overview of what we were looking at is, on the left you will see the S3 bucket where the phase 3 data existed. Within the GenomeNext platform cloud, we were able to consume all the data out of that S3 bucket. From there, we then launched our infrastructure, which houses our analysis platform, which is Churchill. Churchill would then crunch through the data and launch and provision all the EC2 instances necessary. And then when the infrastructure or the instances had finished the analysis, we terminate the environment and we would drop the data into a GenomeNext secure S3 bucket and then present that data back to the end user. So to go into a little bit more detail into the environment, from the left we have we use an Amazon load balancer, which is called Elastic Load Balancing. So a user would be able to access the load balancer, which then hits the GenomeNext portal interface. From there, we have numerous instances running that handle our web front end as well as our database tier. And I would like to note that the architecture that we're using in Amazon is a very decoupled and open architecture, and that different pieces of this architecture can quickly be swapped out as new enhancements to our platform progress. So once the data is uploaded to the GenomeNext platform by the end user and an analysis request is submitted, we actually send the request to Amazon's Q service. And the Q service is basically a container of many messages. In our cases, we had 5,008 messages that was sent to the queue. That queue is being listened on by our Churchill node. And the Churchill node is looking for messages, reads the message. And within that message, we have numerous amounts of information that's in there. One is where the data is located in S3. What type of analysis is this? Is this a whole genome analysis? Is this an exome analysis? And from that information that's provided to the listener then launches the appropriate Amazon machine image, which contains our Churchill pipeline. And at that point, the Churchill pipeline takes over. It launches all the Amazon EC2 instances necessary, and it goes through the analysis. As it's going through its analysis and the pipeline, the Churchill instances communicate back to the queue service. That it's basically our update queue, and that update queue sends messages back to the GenomeNext portal that says, okay, we're at deduplication or we're at this step in the process. Finally, when the analysis is completed, we then are able to terminate the infrastructure and then send a message to the queue that says that the analysis has been completed. And then we also send the data, that's the results data, the variant call file, to the S3 bucket where it is then stored. The user, once he gets the update from the queue, also receives an email saying that the analysis has been completed, can then go back into the user interface, then see the result files and download them directly from S3. So that, from a very high-level overview, is the process that we deployed within Amazon that was allowed us to essentially upload an index file of all of the FASTQ files in the 5008 samples. Once we uploaded it, the process took over, and all of these different pieces of the architecture automated the analysis as well as the automation with the EC2 infrastructure on the cloud. So essentially, per day, we were running somewhere around 500 to 1,000 instances to get through and perform the analysis. Part of the reason we didn't go more than 1,000 instances was we wanted to maintain some control of the environment, and if there were any failures, that we weren't failing on thousands of instances, and that we were able to monitor that closely. In the future, we will have the ability to launch more than 1,000 instances and actually do larger scale population. But for this project, the challenge that we were going through was, can we do it in 7 days? And so we looked at an appropriate amount of instances to be able to accomplish that. In terms of our cloud monitoring, so one of the challenges that we had is we needed to make sure that we knew how much infrastructure was being launched at any given point in time, and that we put a capacity cap at that infrastructure so that when the infrastructure hit the 1,000 instances, that we were able to slow down the queue and wait until new instances came back up so that we could use that pool. And so in order to do that, we built a cloud monitoring solution that was able to look across the different Amazon availability regions that we were deploying instances, check the status of the instance, and when an instance was terminated, that became an available instance that would then relaunch the queue and relaunch EC2 instances to perform analysis. From the UI, we basically have a very simple user interface. So once the analysis has been submitted, essentially we have, it looks like tiles of the different analysis requests, and it gives you a status of what the name of the analysis was, how many FASTQs were associated to it, and what the progress of each analysis was. And we could easily come in and check the status and check to see if there was any issues going on with any of the instances during the process. Additionally, when you go into the analysis and review the sample, we provide lots of information around what the sample was. Is it a whole genome, a whole exome? We provide information about who ran the sample. Additionally, we're able to determine run times of each sample. And we audit the samples to determine how long each step in the process would take. And we were able to do that and track that through time. Additionally, on the top right, once the sample had finished its analysis, we provide the variant call file results, where a user can then download and view the information. Additionally, we link back to the actual sample data. So if you wanted to go in and look through the FASTQ files, you were able to do that as well. So that really ends the presentation on my side for the cloud computing environment and different aspects of the platform. So with that, I'm going to go ahead and hand over control to Peter. I know that we have some, it looks like we have some Q&A that's coming in. Peter, do you want to go ahead and take control? I'm going to go ahead and hand.
Okay, you can, if you have any questions, please feel free to go ahead and submit them through the QA box, and we'll try to answer as many of your questions as we can. I'm just going to look at see what's coming in. So we have a question from Linda. She's asking, you were able to complete the calculations within 7 days. Tell us more on how this impacts the clinical and patient experience, and tell us what's next. Yeah, so one of the things that we've observed from in terms of using the results from the 1,000 genomes analysis is that when we sequence a patient, we use the 1,000 genomes frequencies as a way of filtering out what's a common genetic variant versus a rare, potentially pathogenic variant. And one of the advantages of using the data that we've generated with Churchill was that we could then go back and look if the variant hadn't been called by the 1,000 Genomes Consortium. We were observing that some of the variants we were actually able to call at a fairly high frequency. We believe this is really an effect of doing an apples-to-apples comparison, that when you're filtering your data, ideally you want to use data that's been population frequency data that's been generated with the same method that you use to do your particular study. So I think that's one way that we're hoping to see this sort of impact our ability to more accurately determine whether a variant is common or if it's pathogenic. So we have another question related to the clinic from I think I'm going to mispronounce your name, so I apologize. It's Mikola Razek is asking, what impact has Churchill Bioinformatics had on genome sequencing implementation in the clinic? I think for our clinical group here, the real advantage has been that they've been able to fully automate the analysis process, and it has really removed the need for bioinformatics scientists to become involved. So it's really allowing the clinical staff in the molecular lab to run the analysis and get the results back. So I think that's been one of the major advantages, is just the complete automation of the analysis approach. I'll save that one for James. And then we have a question here, actually, James, I think you would be better able to answer this. Are you able to leverage AWS Direct Connect to upload data to Genome Next?
Yeah, so I'll be able to answer from James Finch. The question is, are you able to leverage AWS Direct Connect to upload data to Genome Next? So there's a couple of strategies around data upload, and the platform for Genome Next implements 3 strategies. One is we do have a solution where a user can use the web interface, upload a sample and the FASTQ files associated to that sample. And in a lot of ways that solution works for some customers, but if you're looking to upload hundreds or thousands of samples from a backlog, that's going to be more challenging through a web interface. Additionally, you're going to have some challenges around bandwidth. If the web interface, if it's choppy or if you don't have good bandwidth, you're going to have some challenges that way. So the other option that we do have is to leverage Amazon Web Services Direct Connect, as well as we can provide isolated S3 buckets to users and give them secure access keys for their organization where they essentially would be able to write directly to a specific Amazon storage location. And in those cases, we essentially allow them to use any tool that they have, any third-party tools. So there are tools out there called like S3 browser. There's some that are pretty inexpensive. Also, there's some tools out there from commercial vendors that allow for data migration to Amazon S3. So utilizing some of those third-party vendor tools, you can quickly upload data. Additionally, the Amazon Direct Connect allows you to have a dedicated line to an Amazon termination point so that you can have a better performance on your bandwidth from your facility directly into Amazon. The third aspect is really, it's also an Amazon service, but we have the ability to allow customers to upload data to drives, and we can then drop ship those drives directly to an Amazon facility securely, and then they're able to upload the data directly from those drives to a secure Amazon storage location that we have access to. In those cases, you know, if you're looking at petabytes of data, probably doing something using Amazon import method that I just mentioned would probably be your best bet. Additionally, got a question from Linda. You did the calculations within 7 days. What was the original target? Was it considered a good result? So the original target was 7 days. In terms of was it a good result? At this point, we don't know of anyone that's been able to analyze that amount of data and that many genomic samples within 7 days. We probably could have done it faster, but you know, we wanted to make sure that it was more of a controlled environment. As I said, moving forward, with the ability to use Amazon's unlimited compute environment and their storage, as well as a lot of the upgrades that Intel are doing to their processor and chipsets, we feel pretty confident that we can actually get that in under 7 days and be able to launch more instances and do larger population scales. With that, I'm going to pass the baton back to Peter.
Okay, well, thank you everyone for listening today. If you have any additional questions, you can feel free to submit them through the LabRoots site. Today's webcast will be available for on-demand viewing for the next 6 months. You'll receive an email from LabRoots alerting you when it's available for replay, and feel free to forward that to any of your colleagues who have missed today's, who may have missed today's live web event. And so finally, I do want to thank our sponsor Intel for making today's educational webcast possible. And thank you all for your attention. Goodbye.